All right. Hello, everybody. Good afternoon. This is Jason Williams from the DNA Learning Center at Cold Spring Harbor Laboratory. Welcome. This is part 2 of RNA-Seq with DNA Subway. So to start, I'm going to go ahead and get some slides up. Let's find where that is. Okay, excellent. All right, so this is part 2 of RNA-Seq with DNA Subway, and this is the DNALC live channel. To prove to you that it's live, I don't know if you hear the sirens in the background, but this is definitely a live show and a live experiment, and we'd love for you to give us some feedback and let us know what you'd like to see. So if you're joining us in the chat, feel free to ask a question. If you are going to leave a comment after the video is done or on one of our social media channels, you can do that. But make sure you go to dnalc.csshl.edu and click on our live site. You'll find, of course, not only places where you can give us feedback there, but like I said, come to any of these other links and here are handles. This is actually an online course, and the audience is for undergraduate biology 200 level and up, maybe at also advanced AP biology graduates, people who have completed that level. So I'm pitching things at that level of audience. If you haven't taken some of those courses, but you're really interested, I hope you get some concepts out of it, but you'd be better prepared if you have at least done that much coursework. The format is 3 sessions, 1 per week. So this is week 2. They're approximately 45 minutes each. I was longer last time. We'll see where I end up this time. You can also follow along using our online bioinformatics tool, which is DNA Subway. So feel free to go ahead and sign up for an account. I'll mention that later. You'll also find that on that dnalc.csshl.edu website, there are slides and resources. So these slides are up there, and there's also a worksheet. The overall course learning goals, just to recap, we want to understand what an RNA-Seq experiment is, the experimental design, understand how DNA sequence works and how to understand the quality, that's important. Then we're using several tools in order to complete our analysis. Last week, we covered most of quality control. And this week, we're going to talk about alignment of reads. And then next week, the last part in our series, we will be looking at exploring the results that we get from the analysis. As I mentioned before, we're using DNA Subway, and you can sign up for a free account. If you Google DNA Subway or go to cyverse.org, you'll find it there. You must have a DNA Subway account to use this analysis that we're doing, since it uses pretty big data, pretty big compute. Okay, so we're going to review some of the key concepts from last time, just so we're all on the same page, and we're going to move into alignment. So I think I've mentioned the first bullet point. The second one is we're also going to talk about where does the data come from that we use to do the alignment, and we'll learn a little bit about the tools, particularly the one that we're using called Callisto. To go back, we remember that an RNA-Seq experiment, the whole purpose of it is to measure the transcriptome. So take an account of all transcription occurring in a cell or maybe even tissues. And we use the abundance of the RNA transcript as a proxy for the activity of some cellular process. So we're just making the assumption that if we see more of an RNA produced, that's going to lead to more of a protein produced, and then maybe that protein has some activity. But of course, there are other things that RNA transcripts might be doing. They may be leading to regulation. There may be other things going on, but essentially we're making that one-to-one assumption. It's not always true, and sometimes RNA-Seq has been called a differential expression experiment. That's a little bit of a misnomer, but it's a good enough approximation most times. The other thing is that we need to analyze, and in an RNA-Seq experiment, we're usually comparing at least one alternative condition to control. So for example, if we're comparing how tissue or how gene expression works in cancer cells versus non-cancer cells, we want to know what are the differences. This was the high-level overview, right? That you need to find out what your experimental design is, whether you have a control group and an experimental group, if there's only one sort of variable you're looking at. Then we need to extract the RNA and prepare it for sequencing in step 2. In step 3, we actually do the sequencing. And then what we're mostly focusing on this series is step 4, the analysis. Just to give you an idea, although this is not actually the results of an RNA-Seq experiment, but this is another way of getting at similar data, that if you look at the electrophoresis results in the figure there in section A, this CYP1A gene in people who didn't smoke, it's not really very much expressed. Those gels look like there's not very much protein there, but in smokers, it is expressed. And in that graph that's in the bottom, you also see that. This is important, or this is what you would expect, because the CYP1A gene is involved in processing toxins. In particular, it's expressed in the lungs very highly in humans. So this is giving you an idea of what gene expression, or getting a readout. In that case, that was a protein, probably a Western blot. But in our case, we're looking directly at the mRNA. So conceptually, that's what we're trying to see, signals like that. We also mentioned last time that when we're doing an RNA-Seq experiment, we need to keep in mind that there's a difference. Well, there's always differences between different biological organisms. And so we wanted to have in our mind the concept of variation versus actual differences. So this is giving you a great example. We're going to come to statistics many times throughout this course in some way or another. If we were looking at a variable like height, this is just a general biological principle. We might see that women tend to be in humans shorter in height than men tend to be. Men tend to be a little bit taller, biologically speaking, about sex. But just because you sample a man and their height, it might be over here underneath the curve that's female, doesn't mean that the sex is mislabeled. It just means that they're on one end of the spectrum, and so on with women who might be taller. So the statistical thing in the RNA-Seq experiment, one of the things that makes it a little bit complex is we need to be able to separate these things out nicely so that we look at gene expression in a cancer cell and we convince ourselves that the gene expression might be much higher or much lower because we're looking at a cancer cell versus it just happens to be that one individual tumor sample was different from another just by chance. Okay, and also, the important thing for our experiment, and we introduced last time, is that in our data set, we're looking at gene expression in human neuroprogenitor stem cells. These are stem cells that lead to many different cell lineages in the brain, except that we're looking at some cells that have been infected with Zika virus, which, as you know, affects, and actually we covered last time, kills those cells. The data is retrieved from NCBI. So there's the sequence read archive, and things that are labeled mock are cells that are not treated with Zika. Things that were Zika are obviously are. And these accession numbers represent the actual data sets. So there are millions of reads, as you can see in the reads column, and we're taking some of that data and analyzing it. I mentioned last time that we use Illumina sequencing to do the generation of sequence data, so go back and watch the video. But one of the most important concepts for this time is the idea of a read. So reads typically in an Illumina sequence are going to be 100 base pairs or more. This is just giving you a little cartoon example, but the idea is that you get perhaps dozens, hundreds, thousands of reads that might cover any section of the genome. We're going to talk about that in a little bit more depth. And you need to kind of look at all of those reads. Sometimes you might have an error like this T. That's probably a mistake since we only have one read showing up with a T. But in some cases, maybe you have half the reads with C and half the reads with A. That may mean everything from you have a spontaneous mutation, maybe unlikely, or if you're comparing different samples, then of course there could be the chance for biological variation. It could be sequencing error, or it could be that the individual is heterozygous for that particular locus. There's lots of confounding factors, and that's what the bioinformatics is about, separating out all those differences and finding the real difference that's interesting versus lots of biological differences or other forms of noise. So last time, and we're going to open up DNA Subway right now, we went into FastX. So we're going to go ahead into DNA Subway and take a look at our results from last time, because last time we started the process, but we didn't finish it because it takes some time. So I'm going to go ahead and share now DNA Subway. Okay. It just so happens that I'm already logged in. And if I go to my projects, if I click the one that we did last time was actually is what called webinar part 1. I'm going to do something a little bit different this time. And we go into manage data. What we did last time was we imported sequence data from all of these stem cell sequences that we generated. And we clicked on view in order to look at the quality of these. And we covered in the last video, go back and watch it if you missed it. We covered what this means. These are actually really high quality sequence because the Phred score is really pretty high. So if I go back for a moment to the slides, I think I have a slide on this. With a Phred score we seeing in the 30s or higher, we're at the point where any given read has only a 1 in 1,000 chance for every base call that you make, 1 in 1,000 chance of there being a mistake, which is pretty good. So that was an important thing from last time. The other important thing was that these individual reads, which I'm going to return back to DNA Subway in a second, are in the form of this thing called a FASTQ file. Now, this is one individual read in a FASTQ file, and there are millions of them in a FASTQ file. This, every entry, if you will, every read has 4 lines. Line 1 is a sequence ID. Line 2 is the actual sequence. Line 3 is this plus sign, and then 4 is this code, which translates to be a number on a scale, which is a Phred score. So let me go back to DNA Subway. And this time, let me open up a project that I continued. Okay. And let's see, is this the correct one? Oh, no, this is the correct one back here. Okay. If I go into the manage data, we went through and we did the QC on all of them. Now, if I click on any given one of them, I'm just clicking on it random, what I think you'll find is that most of the data is really pretty high quality. And what it turns out that you can do is if that data are really high quality, you can move directly to the next step, which is this thing called Callisto. However, if you wanted to, what you could also do is you can go to this next step, which I didn't cover and which I just introduced now, called the FastX Toolkit. So on the FastX Toolkit, this is a piece of software that's been integrated into DNA Subway. And in order to get that to work, what you need to do is I gave instructions to click run for all the basic ones. And one little thing to know about DNA Subway jobs is when you click run, oftentimes you come up with a job number that usually begins with a 2-letter prefix, which is the prefix for the tool, FX for FastX Toolkit. And you come up with a job number, 37937. And those results are available to you also on the Cyberse data store, so you could download them if you wanted to. That's not important just this second, but I think I'll cover it when I get to the Callisto portion. Just so I wanted you to know what that is. When the status is done, you can click view, and what it's going to give you back is another FASTQ report. And it's going to be hard to tell the difference because the sequence quality was so good. But if there were sequences that could be improved by trimming them slightly, or if there were, sequences where it was bad enough that you drop the whole read, you get it. So if I look at this one, I see that I have 7 million, what do I have? Yeah, 7,920,598. And if I click on the same one, let's see if that was really, if I clicked on the first one. Okay, almost the same number, right? But slightly different. If you had poor quality sequence, it's much more important to use the FastX toolkit, but you can skip it if the sequence quality is good. And a little hint that I haven't really written in a slide is that with the method that we're using, Callisto, it's really good and robust to sequencing error. So even you get a little bit of a boost that way. Now to actually look inside of what's happening in FastQ, or rather FastX Toolkit, if you click on Run, what you're going to see is that you have a whole bunch of parameters that you could set in order to change what you get. Now, these columns here are Phred scores, except for the very last one, that you want to filter at. So if you set quality trimmer, minimum quality 20, in fact, it kind of gives you a little bit of a help there. It says that if you have a read and the end of the overall, the average quality Phred score for that read was 20, that's the threshold. So anything less than that is going to be dropped. If you increased it and you said 30, then reads that were less than an average of 30 would be dropped. Once you drop it, or let's say that you have some trimming that's going on, you might say, well, don't give me anything. If after you do a trimming, right? If you go along the read and you decide that it reaches an average and it's like, okay, wait a second, we're dipping below that number, it starts to trim it. But if it trims the read so much that you get, the length to be less than 20, it'll also drop. I think I was not so clear back there. So the quality filter is the overall average for the entire read, and the quality trimmer is just the by position. So when you start getting to positions, it's going to trim the read and shorten it. And this one would just dump the read. In fact, actually, once you have that, this one says if the percentage of bases is greater than or equal to this minimum quality, that's how many. So if you set your minimum quality to 20, 50% have to be greater than or equal to 20 in order to keep that read. So you have this little combination, this little toolkit of manipulations that you can perform. And doing that sometimes will help to rescue and improve the data. And certainly what you want to do is use the highest quality data. So that's the point of the FastX toolkit, to give you those trimming steps. If you have more questions, then I advise you to go ahead and Google FastX toolkit, and eventually you'd come to the Hannon Lab website. This is actually developed at Cold Spring Harbor a while back, but still quite, still used. And there will be additional directions. And I believe those are also linked from the DNA Subway website as well. Okay, let's go back. Okay, so I mentioned, and we went through that. And I mentioned that you get jobs that are labeled like that, so you know what those numbers are. Okay, so the next concept, which we're going to spend some time on, is read alignment. So let me present sort of what I did in an earlier slide, but present it to you in a little bit of a different way to give you a cleaner idea of what the important concepts are. In an mRNA isolation, so let's say we have our cancer sample and our control sample, or in this case, maybe we have our mock cells, our human neuroprogenitor stem cells, and we have the ones that are infected with Zika, and you could say those are sample 1 and sample 2, respectively. We get the RNA out, and then we go ahead and sequence it. Now, when we sequence it, for one individual mRNA transcript, it may be that that transcript, and usually it's the case, is only 100 base pairs long, but maybe the entire, or rather the read is only 100 base pairs long, or 150, or whatever the number is, but the actual transcript may be much longer than that. So you get these little pieces of reads, and that's what we're seeing in the next sort of red box, or what's gonna happen is that if you take any individual read of 100 base pairs or so, we need to match that and find out where on the genome did that come from, and hence what gene does any particular read, how was it generated? And so here we have gene A and gene B. And if you look, there's lots of blue little dashes or individual reads on gene A, and there's much fewer of them on gene B. So the conclusion would be is that gene A is more highly expressed because we see more reads, we observe more reads, and gene B has a lower expression value because there's fewer reads. Now, if you look at sample 2, we have a similar phenomenon. Gene A is more highly expressed than gene B. That's the truth in sample 1. But overall, pardon me, overall, in sample 2, the expression is much lower, both for gene A and for gene B. Now, there's some tricky things because is it is that difference really because sample 1 doesn't have Zika and sample 2 does? We don't know yet. We have to do some more statistics, but it's certainly a hint that there may be a real difference between those two. Now, what ultimately happens is we form counts and ratios and statistical tests and do different things, and we get an idea of what we call fold change, which is that, relative to one sample, gene A and gene B, there might be a threefold change in expression if we count 30 reads of gene A in sample 1, but only 10 in sample 2. That's a threefold difference. And in gene B, there's a twofold change once we do the math. That's what we'd like conceptually be able to do. So this is just summing that up to say, this is the intuition. The more reads that we observe from a given gene, the more active that gene is. And I put the word gene in quotes. I don't just mean protein coding genes. So there could be other things that we're looking at. And also the word active, I'm putting in quotes because again, what we mean by that might be a little ambiguous sometimes, but this sentence overall is still a good one. It's a good intuition to have. Now, the problem is, in order to do all those analyses, this is a figure taken from a paper. There are really many, many, many different ways that you can do. This doesn't even show you the different softwares, which there are maybe hundreds of software packages that you could use to do RNA-Seq. But meanwhile, let me just say that RNA-Seq is quite complicated. There are different steps that you could choose to follow or not. There's just lots and lots of moving pieces. So it's way more, how would you say? It's way more sophisticated than I'm going to give you an explanation, the amount of time that we have here. But, just to sort of know that, don't worry about it. But we're going to focus on some of the essentials so that you'd be better prepared to, and come back to that nuance. So why we've then chosen this software called Callisto is because it's quite simple compared to many of the other software projects out there. And on top of that, it's also quite accurate. So sometimes there's a trade-off where something is more simple and it's less accurate, but actually this has stood the test of time. That's the actual paper, which I believe I provide a reference in the worksheets on our DNLC homepage. But if you Google that, you'll find the paper. Unfortunately, it's a paywall paper, but their website provides some helpful information. So if you go to and search RNA-Seq Callisto, you will find useful information about it. So here are the major steps of doing an RNA-Seq with Callisto. Callisto does something called pseudo alignment, and I'll talk about what that means, but it aligns reads to a reference transcriptome. So transcriptome is a key word here. We'll talk more about it. And there are two steps to what Callisto does. One, it needs to build an index of the reference transcriptome, and I'll describe what that means in a bit. And then two, the sequence reads need to be pseudo aligned. So number one, you can do without any experiment. Number one, as you're going to see in a moment, we can just go to a website and find the transcriptome of our organism that we're interested in. That's easy. Number two, we actually need to do the experiment to generate reads. And then once we have those reads, we need to align them to actual transcripts that we get from step one. So to do that, what I want to take you to now for just a moment is where do we get the transcripts, and what is the transcriptome? Well, I put here a transcriptome is a collection of all, and I put that in quotation marks, all the transcripts of an organism. I put that in quotation marks because gene expression happens in really sophisticated and well-controlled ways. So for example, if I want to ask if I wanted to know all of the possible transcripts in a human being, humans have somewhere a little over 20,000 genes, somewhere between 20 and 25,000 genes. That is many times, maybe 5 times lower than when the Human Genome Project was done. When we first sequenced the human genome, we knew of more than 100,000 RNA transcripts from cDNAs. We knew there was lots of RNA and lots of different transcripts. So we were surprised that there were so few actual genes. And turns out that, most genes have different isoforms, so different The transcript can look differently depending on different circumstances or different developmental time points. Um, so if I go ahead and I sample, let's say, blood from a human being, there's no way I'm going to get transcripts that are only expressed in the liver or only expressed in the eye. I'm only going to get a blood transcriptome. So you have to really sum up all of the transcriptomes from all of the tissues, from all of the organs, and all of the developmental time points in order to get a complete transcriptome. That may not be possible. There are new transcripts discovered all the time. Human, it's harder because it is a well-studied organism, but in lots of organisms, maybe your transcript is not represented in the transcriptome. That's yet another thing that's a problem. But for right now, let's go ahead and take a look at the human transcriptome and where you get that data, and then we'll be able to continue on from there. So I'm going to go back and share. And I could have pulled the link directly from there. The link is in the worksheet, but what I'll do is I'll just Google Ensembl. There was my last search, right? Ensembl human genome. So this is the Ensembl website database, and Ensembl is a large project that houses many, many interesting data types for humans. If I were to click on this thing called example region, it's going to open up a genome browser. And it looks like I am on chromosome 17. And on chromosome 17, I can see all of these little names. That's interesting. I don't know what that gene is. There's a gene called TACO1. It's almost towards lunch. So let's click on jump to that region. And in fact, if I click on the plus button, let's see if I can zoom in a little bit. Give it a moment to hopefully load in. Whoops. What I wanted to do, I can put a marker there. You can go ahead and it's going to zoom in down here. And what I see are actual, this is this particular gene here with boxes representing exons and introns representing lines. But these are, various genes that are somewhere in the human genome. And for any particular gene, if I click on that there in this sort of more detailed view, it's going to pull information. So this is, oh, that's a cute name, translational activator of cytochrome C oxidase. So I guess that stands for TACO. I didn't know that. But if you go up, if you look here at what we're seeing, these are ESTs, but what we are actually getting, these are essentially equivalent, cDNAs. These are basically equivalent to individual transcripts that are like RNA-Seq reads. And in fact, there are probably some, this is very much what an RNA-Seq read looks like, where you have to imagine that this lighter part is an intron, and then you get actual reads where it's an exon. And there's all various things. Now, this is cute, this is nice. And if I have a gene that I'm interested in, I certainly want to come back and view it in detail. But this data, I need to get it all at once. So what I actually need to do is come back to the human genome here and then say that I want to download some files. So if I click on download FASTA, which is a file format, and then I say I want to get all of what we call the cDNAs, which are the transcripts. These have been generated by an experiment. And if I click on any one of these, I'm not going to click on it. Maybe I will. Is it a dumb idea? Let's just do it. I'll click on this smaller one, which are predicted cDNAs. I'll try to open it. Probably it can open in a text editor. I'm going to have to share that. Give me a second. Text edit. Let's see if this is a smart idea or not. Okay, not too bad. Let's share this. So this really long text file, that's all that it is a listing of all of the actual real transcripts that are in the human genome. So, here is one individual transcript from one gene, and it gives you the gene number. Um, and you'd have to go back and look that up to actually get a name for these genes. And you can see some of the transcripts are longer, some of them are shorter, but these are the actual pieces of the human genome, the actual protein coding ones, or cDNAs for the most part. So we need this. And to sum that up, going back to where I was just a moment ago, to sum that up, this Ensembl database that we were just visiting is where we want to go in order to download the reference transcriptome. And I've just looked at human, but you can go ahead and search, any of these organisms that are here. You'd find transcriptomes. If I click on, back on that database example transcript, it's just going to take me to a very similar view to one that we had before. Taking a long time to load, so I may not may not go back to it completely. Let me go back to this. It's more interesting. Yeah, here's one of the FOXP genes, and again, these are individual transcripts. So the alternative would be to download the entire human genome. We're not going to do that. We could do that. And in fact, actually, some RNA-Seq programs will align to the entire human genome. But if you think about it, we don't need the introns, right? And we don't need large sections of the genome where there's not any gene expression going on. It's true that gene expression is not confined to protein coding genes, but we could skip it for the sake of what we're doing. Just wanted to go back to the README, and it gives you a little bit of an information about what you're looking at. And it's useful to view that sometimes. We don't have to worry too much, but it tells you that the cDNA sequences, where they came from. It gives you what's the difference between all or an ab initio, et cetera, et cetera, et cetera. So you can go and look in that. And it also decodes, what is the name, how are these things named? So you can go back and look that up. Okay, so let's go back to our slides and back to DNA Subway and the work that we have to do. Okay. So there's a problem. One of the problems, as you saw in just that file that we're looking at, that it contains thousands of transcripts. So the question is, how will we match sequence reads with transcripts? We get millions of sequence reads, and we need to find out which transcript did those reads come from. And this is really a big puzzle. And it's kind of a messy puzzle because in that text file, those reads are, for all intents and purposes, random, or rather those transcripts. So the question is, how do we sort through all of those transcripts? Can you imagine having millions of reads and needing to align them to thousands of transcripts? That's a gigantic puzzle. You have to take each one of those individual reads that you get, let's say, from your Illumina sequencing, and then find where on the reference genome, or in our case, reference transcriptome, do they match. You have to deal with the fact that some reads will match more than one gene because lots of genes are homologous and they share nucleotide sequence. So you'd need to, figure out what you do with that. There's a dozen problems to doing that. I can't talk about them all, but just let's know that it's a complex process. So one of the things that you could do in order to make that process much faster and less complex is you could try perhaps to sort the puzzle. You could sort those transcripts in a way that makes sense and makes them easier to search so that you could have a better time, matching and solving the puzzle. And this is sort of what it looks like. So here is a view. On the top, we have the DNA, or in this case, it could be our transcripts laid end to end. And then each of these red and blue bars are individual reads. And one thing that you might notice is that those reads will overlap many times, kind of like we saw in one of our earlier diagrams. And what you end up with is needing to map them to the DNA that's on top. Anywhere where those reads might contain either sequencing errors or natural variations are marked so you actually see the nucleotides. Like this read had an A here, but that's not what the reference says. Now, there is a sophisticated concept from computer science that allows you to do this type of mapping and sorting and figuring out where things go. And it's called a de Bruijn graph. This is what it looks like for a computer scientist, perhaps. Whoops, I'm getting ahead of myself. Where you ask the question, given a set of reads, if I look at those reads and try to map them to a gene, in what places do they map and what places do they not map? Now, it turns out there's a little bit of a secret, which is that if you take those transcripts and not only sort them, but also chop them into smaller pieces, which we call k-mers, you actually are able to do this mapping. This is pretty sophisticated thing to explain in just a few minutes, but let me try to give you a little bit of something that can help you understand some of this, and then you have to go do it on your own. So there is a nice way to explain what a de Bruijn graph, or a nice analogy here. And it's actually really more than an analogy, because this is the actual mathematical problem that came from it. And this math problem is one that's a little bit easy to understand. Because in math, there is something called a graph, or graph theory, which explains how things are connected, or how you connect different pieces. And in this original thing, there is a town called Königsberg. And in that town, on the river, there's actually a little island. And there are all of these bridges. Actually, there's, I believe, 7. 1, 2, 3, 4, 5, 6, and 7, kind of highlighted in blue. And somebody had the question one day, and this is the type of thing that mathematicians like to do, is can I visit all of the bridges? Can I cross all of the bridges? Is there one path that will allow me to cross all of the bridges only once without going over the same bridge twice? So can I go, should I start from this bridge and then go to that one and then come over across here? But now, if I want to come back around, I've got to come down to here, go over this one. But if I want to come back to this very last bridge, I either have to come back against this one again, or I have to go over that one. So I'd end up visiting those two bridges twice. And my goal was to find, is there only one bridge that I could look at? So it turns out that there is a way of representing this that looks a lot like, or ends up being a lot like, a genome assembly. And what we could do is be able to assemble that genome. Now, before I go on to that, let me also address the question in the chat, which we have. It's slightly related, but not completely on topic. When we have an alignment, the question is asked, if you're making a comparison between protein sequences, can you do an alignment and do a phylogenetic tree? And you can. This is not necessarily the same problem, but go back and watch the other series on Blue Line Bioinformatics, particularly part 3, and we talk about doing phylogenetic analyses and forming trees using sequences. We talk about DNA sequences, but in principle, protein sequences apply, and you can do that with DNA subway. So back to this idea of graphing, here is a sequence, which could be a read that we get from an Illumina sequencing machine. And what we do in order to make a graph and ask the question, is there a way where we can assemble those reads so that they uniquely map and cross only once, or the fewest number of times? What we need to do is take that sequence and chop it up into small pieces, which is counterintuitive. Well, that's like trying to say, well, if I want to solve a puzzle, can I, make it easier to solve by chopping up the puzzle pieces into even smaller puzzle pieces? But when you're working with sequences, in our case, DNA sequences, it works that you chop them up into these overlapping pieces, and each chunk, or each piece, which we call a k-mer, if you choose a particular number for K, like 7, then it's called a 7-mer, or if you chose 8, it would be an 8-mer. Well, actually, it would always be an odd number. We do that for a reason. But you chop it up, and then you say that this piece is connected to this piece, is connected to this piece, and so on and so on and so forth. And you get this thing called a graph. And what this allows you to do is, if you have two pieces that look pretty similar, but you want to map things to them, you actually can resolve really nicely whether, at some point, chromosome 1 and chromosome 2 look pretty different, but then they actually share a point where they look the same, and then they go off and they start looking different again. It turns out that this de Bruijn graph method, which I'm not giving you a detailed explanation for, it's beyond the scope of what we're trying to do here, but you should at least know the term and maybe even look into it. It turns out that this method of building a graph and asking the question, how many times can I uniquely pass through this sequence, is related to that bridge problem, and ultimately allows you to graph or build an index for the entire transcriptome that makes it really easy to search and makes it really easy to have comparisons where you're looking for what I would say is the most parsimonious comparison, the comparison that allows you to make that match from the reads to the actual transcriptome in our case. So that's the problem we're trying to solve. If you remember nothing else, even if you're not really even interested in the math behind it, the problem that has to be solved, and which great mathematicians have thankfully solved on our behalf, is how do we make that searching more efficient? Because if you're trying to map 1 million individual reads to thousands or tens of thousands of transcripts, very quickly that becomes something that's more computationally complex than most conventional computers will be able to handle. Now, this figure is actually taken from the paper. I'll go into it a little bit, and then I'll give us some analogies, and then I'll take us through the last part of today. We're actually going to do the alignment. If you imagine in black here that we have a transcript, that's this thing in black. But if you also imagine that we get a whole bunch of reads, and they're different colors here, pink and blue and green. In B, C, D, and E, what we actually have is the de Bruijn graph. And what you're asking the question is if I look at these reads, which one of these reads could have generated that transcript? Now, it actually, here's another way to think about it. Here is a read that looks like this at the top here, and here's the genome. And we have transcript 1, transcript 2, transcript 3. If we try to do a matching, it turns out that, they all start UCA, so you can't tell the difference. And then they all continue on CAA, CAA, CAA. But when you get to this GU, our read, which in back in DNA land is GT, but in RNA land is GU. Well, transcript 1 and transcript 2 both have that GU or that GT, but transcript 3 does not have it. Therefore, you can conclude that transcript 3 could not have generated this read. And so this read could map to transcript 1, it could map to transcript 3, but we can eliminate transcript 3. And the whole thing that Callisto is doing in the process of the software is it's looking at every transcript and looking at every read and saying, what are the possible transcripts that could have given rise to this read? And in the best case, it'll be uniquely that some read uniquely maps to one transcript, but in many cases, it'll be ambiguous. And so you get these bubbles of where there's some ambiguity. But basically, you get this phrase, you get this case where you try to figure out what reads and what transcripts could match. Now, to make that even simpler, and why we call this pseudo alignment, is in traditional alignment, which I didn't want to go into, you try to do that for every single nucleotide, and that's expensive. But the really great insight of the developers of Callisto was that we don't need to look everywhere on the transcript. If we actually just sample, and these are these little circles in black, if we sample the transcript at different intervals, if we see it matching, then we could say, okay, it's possible. It could still be true. That's kind of like, if you're playing a bingo or you're playing a lottery and somebody calls out the numbers and the first number is 2, or raffle is a great thing. And you have a 2. So you're like, okay, I could still win. And they call it a 3 and you have a 3 and you're like, all right, I could still win. Then they call it a 5 and you have a 7. At that point, your ticket is not a winner, all the way up until it didn't match. So what you do here is you sample it along the path, and where it's not a match, then you realize that that transcript no longer could match. And if it continues to be a match, then you keep it. Here is another analogy, and it's an interesting one. I'm going to give you one more. Sorry, I'm giving you all these analogies, but I think maybe you have to think about this in certain ways or multiple times, and one of them might click. Can you read this? If you can, you're probably a native English speaker, or at least have native English fluency, but it's interesting. I can read it. Let me try. I couldn't believe that I could actually understand what I was reading using the incredible power of the human mind, according to research at Cambridge University. It doesn't matter in what order the letters in a word are. The only important thing is that the first and the last letter be in the right place. And that's pretty amazing. All of these words here, which probably you can read at with context, that you could probably read pretty naturally. Um, the first letter and the first and last letter of every word is the same, but every other letter inside the word is completely scrambled and random. But the point is, in this analogy, is that sometimes we can be more computationally efficient if we don't have to consider all the data or all of the read. We can just look at a part of it, and if the read is matching at that part, we keep looking a little bit harder. But the moment we realize it can't be a match, and we toss it away and say that's not an alignment. There's another analogy, my last one, I promise, but I also thought that this helps to give you, there's a game that I had just recalled when I was thinking about this that gives us the idea of when we're doing this thing called pseudo alignment, we can look at just part of the read and figure out if it came from any particular transcript. So this is a game. You could actually go and Google it and play it. And it's a cool game based upon Google Maps. And what they do is they show you a photo. Here is a photo. And you can normally scroll around a little bit. I'm not doing it here because it's not live. But the question they ask you is, where in the world is this photo taken? So give you a second. Where in the world, where on the globe? This photo could literally be from just about anywhere on Earth. I'm sure Antarctica is underrepresented, but where on Earth do you think this photo was taken? Okay, so you can think about it. You can pause the video and come back to it, but that's it. Now, the thing that you do next is you actually pull up a map. I hid it just so you didn't see this at the first point. And then on the map, you can put a pin to where you think that that photo might have been taken. Now, when I looked at this, it looks like it's kind of semi-tropical. It doesn't seem like I'm in a city or someplace that's really, really developed. It also, just in the clouds, it looks muggy and hot. So I thought that, you know, maybe this is somewhere in Southeast Asia. So you put your pin down and then you guess. And in this case, I put my pin down here like in Vietnam, but it was kind of in Singapore. So my guess was off by 2,000 kilometers, 1,200 or so miles. And you get a number of points. And the closer you are to the real place, the better you get. So what about this next photo? Where is it? Okay, I'm looking, and when I look for things like that, I try to look at what kind of car style do they have, the roads, and when in this case, I looked around and I saw that there was some chalk on the ground, and it's like, okay, that looks like Cyrillic. So maybe this is sort of like in Russia or somewhere like that. And in fact, I put the pin somewhere in Western Russia, and in fact, it was in Ukraine near Odessa. It's just like in a city. I should have put it right in a city, right? And this is much closer. I was only off by 600 miles. My point in this analogy is that when we are doing the secret of Callisto as a computational tool is that we don't need to look everywhere. We can infer lots just from looking at a little bit of the data. And what this does is that, although I didn't include the slide here, this method, Callisto, can take on the order of a few minutes, where previous methods took hours. So just by looking at the right part of the data and just a little bit of it, we can advance what we're able to do. So we're going to go in and look at pseudo alignment. I'm going to explain this and show you what you get, and then we're done for today's lab. Let me go back to share. And let's go back to DNA Subway. So back on DNA Subway, we can close this. And the next step that we need to go to is Callisto Quantify. Now, I've already run some jobs, and we're going to take a look at those results. But what you would do is you would say, submit a job. And what you'll be presented to when you're asked to submit a job is our DNA sequence reads. And you have a left read and you have a right read. And if you did FastX Toolkit, you're going to have more than one here. So in my case, although I think it's mislabeled, you get this thing called Top Hat, which I believe is my trimmed reads. And then you get ones that are untrimmed by FastX. So you need to choose how you're going to both name these samples and what condition they were. So this information is also linked to you in the worksheet. If I go back and share It's also in my slide. This is taken, whoops, this is taken from the worksheet. The 4.2 samples are mock 1-1. That's the sample name, but the condition is mock. So you can imagine that we have some replicates here. In this case, we've only got 2 replicates. The absolute minimum, it would be better to have more if possible, but that's okay. But this is just so you know what to fill in on this if you're doing it from home. And what I would recommend is that if you're watching this video, go ahead and do it so that you're prepared for the next video according to this chart. And that's also on the DNA Subway guide at learning.cybers.org. Let's go back to Subway and enter this. So I believe it's going to remember. I'm going to say that this is Mach 1, Mach 2, 1. Thankfully, it remembers, so I don't have to type this up. These are both Mach, and this is Zika. Okay. Now, when you run this and click submit, I'm not going to click submit, but when you run this, it'll go ahead and do that job, and it takes about 30 minutes. There are also some parameters, which I'm not doing. So these you can look on the Callisto website, but by and large, we don't need to touch these, and I'm not really going to explain any of them for today's thing, although maybe the bootstrap is important to know, because when Callisto does that matching, there are some probabilities involved and some sampling involved. And so it does that sampling multiple times. That's what a bootstrap is. And you'll see that later when we look at the gene counts, which is what this step is designed to give us. What I am going to do is close this and look at our results. So when you're finished with Callisto, what you end up getting for every time you launch it, you get a job number. So those are these tabs here, and you get some information. Now, if I click on the little number here, it tells me how many reads there were in my sample. So in this case, when I compare, when I add all of them, there's 7.5 million reads. And it tells you how many of those were actually pseudo-aligned. So if I actually look at it, about 2 million of those, there's an alignment rate of only 30%. If I go back and let's look at the other job, because I haven't looked at the results from those. Yeah, I think that this, the other job, doing it, choosing not the top hat samples, but the ones that are just regular, gave me a much better alignment rate. So I would expect that somewhere along 80% of my reads actually map to my reference transcriptome. So this is definitely the data that I want to take forward. The other thing is that if you click on the little folder there, it's going to take you to Cyverse, which I haven't explained very much. I happen probably to already be logged in, but this is a much larger project that we at the DNA Learning Center are involved in. The reason why I wanted to go to that is because it's going to actually open up a folder where those data are. And if you are looking at any experiment, let's go back. What job number was this? This was 38025. So if I look at that job there, it's going to have a whole bunch of things, including my CSV file with some of my reads. I don't need to look at it here per se, but it is going to be here. And this is getting more into next week's, so that's pretty cool. But let me go back to DNA Subway. What I wanted to show you is that if you click on the download here, let's just click on one of them. It's going to open up a great big spreadsheet. And let's see. I think I'm going to download. Let's see if it will go ahead and open up in Excel for me first. And in fact, these spreadsheets are so large that it can have a difficult time even opening in a program like Excel. So if it doesn't work for you, you can choose to just continue with DNA Subway to the next stop. But I'm going to try and open it. Okay, so it gives us this spreadsheet. And let me share it so you could see it. This is setting us up for next time because I'm not going to explain this one so much, but let me give you the first bit of it and we'll go into it more. So each one of these targets is actually one of our transcripts in the human genome in our case. Length and effective length, I'm not going to talk about this second. That'll have to do with next week's exercise, but let's scroll a little bit. The two values that are on here are the number of counts. So this is how many times a read that we got from our experiment matched to this particular gene, or this particular transcript. So for example, in this transcript, which has a long label, but not really a name that we know right now, we'll get that later, had 0 counts. So there was no reads. This was not expressed in any of our samples, or in the sample that we're looking at. And so there's nothing to look at. But if we scroll down and find one, okay, it looks like there are 122 counts, or 122 times that we observe reads. And you'll notice that this is decimal. The reason for the decimal is because we did that bootstrapping where we sampled things multiple times so we get an idea based upon the variation of what the true number might have been. And the final thing next to it is this thing called TPM, transcripts per million map reads, and that's normalization, which we'll explain. But let me give you a quick explanation, which is if a transcript is really long, we expect that there are more reads just for the fact that it's a really long transcript. And a really tiny transcript that's short might not have that many reads, but it's also shorter. So you need to account for that effect. And basically, if you're looking at this, it's a normalization. So saying we can say that the count is actually 20. What we need to do next time is we need to come back to all of our samples. And once we have this count data, we need to look at the count data for our first replicate of the mock and our second replicate of the mock. And then we also need to compare that to our replicates for the Zika. And when we do that and do the statistics of it, that is going to allow us to do the differential expression analysis and abundance. So that's what is for next time. So make sure you go and look at DNALC live, and you'll see the next event, which is about the same time next week. Now I see one question in the chat, and I'll answer that. And if there are more questions, you can please leave them in the comments. The question is something about whether we can use pseudo alignment to analyze DNA sequencing or not. And I guess it depends on what you're talking about DNA sequencing. Really, the pseudo alignment strategy is specific to RNA-Seq, where we don't need all the information about how reads align. We just need to know whether a transcript could give rise to a read. If you're doing a genome assembly, then we would use another method. I don't know if that's your question, but that is hopefully something that gives you a little bit of a clue. If you have more than that, then please come to us in the comments. So it's been closer to an hour. I thought maybe I would try to get to 45 minutes, maybe next time, but there's so much to explain. And hopefully this gives you a little bit of an insight into what RNA-Seq is. So if there are further questions, leave them in the comments. Take a look at the worksheet on our website. And don't forget to like, share, and subscribe so you know when things are coming out, and definitely have access. So with that, thanks very much for attending this presentation, and I hope you have a wonderful day.